Imagine a world where we could rewrite the code of life itself. Genetic diseases eradicated, extinct species revived, and crops thriving in a changing climate. Thanks to CRISPR, this future is closer than we think. For centuries, humans have manipulated genetics through selective breeding. Traditional methods are slow and imprecise. CRISPR offers precision and control over genetic material. This technology could revolutionize medicine, agriculture, and our understanding of life. CRISPR is ushering in a new era of genetic engineering. It's essential to understand the power and implications of this technology. Let's explore CRISPR's potential to reshape the future of life on Earth. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, might sound like a mouthful, but its function is surprisingly simple. It acts like a pair of molecular scissors that can cut DNA at precise locations. This revolutionary technology was first discovered in bacteria, where it serves as an immune system against invading viruses. Here's how it works. CRISPR utilizes a guide RNA molecule which acts like a homing beacon to target a specific DNA sequence within a cell. Once the guide RNA identifies its target, an enzyme called Cas9, the molecular scissors, binds to the DNA and makes a precise cut. This cut disables the targeted gene or allows for the insertion of new genetic material. Think of CRISPR as a sophisticated word processor for DNA. It allows scientists to edit the code of life with remarkable precision, correcting genetic defects, inserting beneficial genes, or even activating dormant ones. This newfound ability to manipulate DNA with such accuracy has opened up a world of possibilities, from developing groundbreaking medical treatments to resurrecting extinct species. De-extinction, the concept of bringing extinct species back to life, was once relegated to the realm of science fiction. However, with the advent of powerful gene-editing tools like CRISPR, what was once a distant dream is now becoming a tantalizing possibility. Scientists envision a future where CRISPR could be used to resurrect extinct animals by editing the genomes of their closest living relatives. By strategically modifying the DNA of these modern-day counterparts, researchers could essentially rewrite the genetic code to resemble that of their extinct ancestors, potentially bringing back creatures lost to time. The prospect of bringing back extinct species is as awe-inspiring as it is controversial. While the idea of seeing a woolly mammoth roam the tundra again is captivating, it also raises profound ethical questions about our responsibility towards the natural world and the potential consequences of tampering with the delicate balance of ecosystems. Imagine a pack of dire wolves, their imposing figures silhouetted against the setting sun, their haunting howls echoing through the ancient forests. These iconic predators, larger and more powerfully built than their grey wolf cousins, once roamed North America, their remains often found alongside those of their prey, the megafauna that populated the continent during the Pleistocene epoch. Dire wolves have captured our imaginations for centuries, inspiring legends and captivating storytellers. Now, thanks to CRISPR, the prospect of bringing these magnificent creatures back from extinction is within the realm of possibility. Scientists believe that by using CRISPR to edit the genomes of modern-day grey wolves, they can essentially re-evolve the traits that characterize their extinct relatives. By focusing on specific genes associated with size, bone structure, and other key characteristics, researchers could potentially create a lineage of wolves that bear a striking resemblance to their extinct ancestors. This ambitious project highlights the potential of CRISPR to not only resurrect lost species, but also to help us better understand the evolutionary processes that shaped life on Earth. Section 5. Woolly mammoths, giants of the Ice Age return. Perhaps no extinct creature captures the imagination quite like the woolly mammoth, a behemoth of the Ice Age. These majestic creatures once roamed the frozen landscapes of the Northern Hemisphere. Bringing back the woolly mammoth is a flagship project for de-extinction efforts. Scientists are exploring using CRISPR to edit the genome of the Asian elephant, the mammoth's closest living relative. The idea is to create a mammoth-like elephant for the Arctic tundra. They could help restore degraded ecosystems and mitigate climate change. While seeing herds of woolly mammoths is still years away, the research today highlights CRISPR's potential to reshape our world. Section 6. CRISPR, advancements fueling the de-extinction dream. Recent advancements in CRISPR technology have brought the dream of de-extinction closer to reality. One significant breakthrough is the development of more efficient and precise CA's enzymes, the molecular scissors that cut DNA. These improved enzymes allow for even greater accuracy in gene editing, reducing the risk of unintended consequences. Another exciting development is the emergence of prime editing, a more advanced form of CRISPR that allows for even more precise and versatile genetic modifications. Prime editing acts like a molecular word processor, enabling scientists to insert, delete, or replace specific DNA sequences with unprecedented accuracy. These advancements are fueling the de-extinction movement, providing scientists with increasingly sophisticated tools to manipulate genetic code. As CRISPR technology continues to evolve at an unprecedented pace, the possibilities for resurrecting extinct species and shaping the future of life on Earth are becoming increasingly boundless. Section 7. A future shaped by CRISPR beyond de-extinction. While the prospect of bringing back extinct species like the woolly mammoth and the dire wolf is captivating, the applications of CRISPR extend far beyond de-extinction. This revolutionary technology holds immense promise for revolutionizing medicine, agriculture, and conservation efforts worldwide. In the realm of medicine, CRISPR is being explored as a potential cure for genetic diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease. By correcting the faulty genes that cause these debilitating conditions, CRISPR could usher in a new era of gene therapy, offering hope for millions of people around the world. Beyond treating diseases, CRISPR could also be used to enhance crops, making them more resistant to pests, diseases, and harsh environmental conditions. This could lead to increased food production and reduced reliance on pesticides and herbicides, making our agricultural practices more sustainable and environmentally friendly. Section 8. The ethical frontier, navigating the dilemmas of de-extinction. The prospect of bringing back extinct species raises profound ethical questions about our relationship with the natural world and the potential consequences of our actions. While the idea of resurrecting lost creatures is undoubtedly captivating, it's crucial to proceed with caution and engage in thoughtful ethical considerations. One primary concern is the welfare of the resurrected animals themselves. Introducing a species back into an environment that has changed significantly since its extinction could have unintended and potentially harmful consequences for the animals themselves and the existing ecosystems they inhabit. Furthermore, some argue that focusing on de-extinction could divert precious resources away from conservation efforts aimed at protecting endangered species currently at risk of extinction. The resources required for such ambitious projects are vast, and it's essential to ensure that these efforts don't come at the expense of protecting the biodiversity we have today. Section 9. A new dawn for conservation, CRISPR's potential to rewrite the story of life. CRISPR is a powerful tool with the potential for both incredible good and unforeseen consequences. The potential benefits of this technology for conservation are undeniable. CRISPR could help endangered species adapt to climate change and resist diseases. By enhancing genetic resilience, we can increase their survival chances. The story of CRISPR is about our evolving relationship with the natural world. We must proceed with caution, guided by ethics and respect for all living things.